Adrenaline Signaling Pathway in the Liver and Muscle Cells by
Taichi Okamura.
and Chloe Lin in Period 3. Adrenaline, or epinephrine, is a signaling molecule that humans can produce, which regulates the release of glucose from the liver to act as fuel for muscle cells. We will get into the specifics about the responses later. Epinephrine, or adrenaline, is commonly known to play a role within our body's flight or flight response. Is the signaling molecule hydrophilic or hydrophobic? Adrenaline is a polar hydrophilic molecule which prevents it from being able to enter the cell through the lipid membrane. To counteract this, a surface protein called the G protein coupled receptor binds to the adrenaline molecule and starts the epinephrine signaling pathway within liver and muscle cells.
Take.
What is the group of receptor proteins typically used for in the cell? G protein coupled receptors in adrenaline are typically responsible for mediating most cellular responses to hormones and neurotransmitters, in this case, adrenaline. Now, for advantages and disadvantages of adrenaline. Advantages of G protein coupled receptors is that these receptors mediate the majority of our responses to hormones and neurotransmitters and environmental stimulants. This means that for scientists, GPCR are worldwide drug targets. GPCR are also great drug targets because binding sites are located outside the cell membrane. 30 to 40% of prescription drugs target G protein coupled receptors, allowing them to be very quick and have short responses. For the disadvantages of G protein coupled receptors is that the actual development of drugs that bind to GPCR is very difficult. It is also very difficult to determine the structure of G protein coupled receptors because of their instability.
How does the GPCR work in general terms? After the binding of a hormone, the G protein coupled receptor undergoes a conformational change where a G protein binds to the receptor. The conformational change is like this. The G protein binds like that. The G protein is split into three parts. There's an alpha particle, a beta particle, and a gamma particle, or subunit, sorry. And the alpha subunit carries a GDP molecule, which acts as a energy source because after binding, the GDP molecule is replaced by a GTP molecule, which activates the G protein. After activation, the alpha particle, beta particle, and gamma particle split up, and only the alpha particle, which carries a GTP molecule, wanders off to an inactive target protein, which after binding gets activated. And this activation triggers a response, and some of these responses can be amplified by specific enzymes called kinases, which are activated by cyclic adenosine monophosphate, CAMP. Kinases, once activated, phosphorylate other inactive proteins to activate them over and over again, creating a large cellular response. That is a cascade and is a very good example of amplification. Alright, here is our detailed explanation of the adrenaline signaling pathway. For the adrenaline signaling pathway to start, there has to be a stimulus, which is either physical or mental stress applied to the host. So we have a character here which is scared by this ghost and is now stressed. Once the host is under stress, their adrenal glands will produce a hormone called epinephrine or adrenaline, which are the same thing, and they bind to the G protein coupled receptors, which we went over earlier. Once again, the G protein coupled receptor undergoes a conformational change like this and binds to a G protein. This G protein is split into three subunits, and the alpha subunit, which is the GDP carrier, changes into a GTP carrier, which activates the G protein alpha particle, and from there it activates our target protein, which in our case is adenylyl cyclase, which is an enzyme that converts ATP molecules into cAMP, which is cyclic adenosine monophosphate. cAMP is a messenger molecule that begins the cascade of multiple proteins getting phosphorylated, which starts at the protein kinase, becoming active protein kinase, which activates the inactive phosphorylase kinase, and this active phosphorylase kinase activates the inactive glycogen phosphorylase, which is the final enzyme of this cascade, and finally breaks down glycogen into glucose. This glycogen is stored inside of the liver, and once it gets broken down into glucose, it is released into the bloodstream and used by muscle cells to use as fuel and metabolize to help our character activate his fight or flight response. Okay, the intended response of the signaling pathway. The intended response of adrenaline signaling is the release of the hormone epinephrine, which binds to a G protein coupled receptor, which is located in the liver and muscle cells. And there are multiple of these cells there are multiple of these receptors in different parts of the body, but in our presentation, we are just going to cover the liver and muscle cells. Upon receiving these signals, the liver cell breaks down glycogen into glucose, which is released into the bloodstream and allows muscles to metabolize and create a surplus of ATP to once again fuel their fight or flight response. As long as the epinephrine is binded to the G protein coupled receptor, the receptor will further activate the G proteins and continue to break down glycogen into glucose. How is the signaling pathway terminated? There are actually two methods of termination. The epinephrine signal transduction pathway can be terminated by self-inactivation of the G protein, and it does this by taking a water molecule and hydrolysizing the GTP that is binded to it. And yes, under hydrolysis, the GTP turns into GDP, inactivating the G protein as well as the target protein adenylyl cyclase, which in turn reduces the production of cAMP. The other method is where the epinephrine that is binded onto the binding site slowly dissociates and causes the entire pathway to stop.
An explanation of a specific antagonist of the signaling pathway is propanopel, which is a beta adrenergic antagonist, aka beta blocker, as it blocks epinephrine at the receptor binding site. Propanopel, an antagonist towards adrenaline, is a beta blocker that is a lipid solution and can cross the blood barrier. It works by responding to nerve impulses and blocking the effects that are caused by adrenaline, such as fast heart rate, sweating, and shaking of chemical messengers. Propranolol is a synthesized drug also known as an exogenous antagonist, which became patented in 1962 and is commonly used as a medication in the US. Now, propanopel blockers compete with sympathomimetic neurotransmitters to the receptors, which inhibits the stimulation of the sympathetic nervous system of the heart. Due to the blocking of neurotransmitters which are binded to the beta blocker, the synthesis of cAMP catalyzes the reduction of protein kinase, or PKA. This results in a decreased effect towards heart rate, blood pressure, while the blockage of neurotransmitters that bind to the beta blocker on the muscle cells will cause an increase in contraction causing hypertension. Overall, it reduces the effects of adrenaline in the body. Now, how the cell response is altered due to the antagonist propranolol is it's altered because the initial response for adrenaline is terminated by reducing the production of cAMP and the activity of protein kinase A.
Thank you very much.